Welcome everybody. Today I continue my review on the bacteriophages and their application in the biotechnology, and we are going to talk about phage display systems, how these systems can be used in the construction of biosensors and how phages can be used in the construction of biosensors, and also we're going to take a glimpse at what is biocontrol with the use of phages. But we are going to talk about biocontrol on our next and final meeting. So, about phage display, as already mentioned, not only classical phages that have head, tail, and fibers can be used in biotechnology, but also filamentous phages. They are like flexible rods. They can be also used widely, especially in phage display systems, because these phages have larger surface area, and these proteins, phage proteins, can be used to express our proteins of interest, new proteins, and this is called phage display. It means that the phage displays something on its surface, displays some proteins or peptides of interest. How this phage display system is created, how it works. It was first introduced in 1985, and now it has a very wide usage in the field of biotechnology and medicine and in laboratory use everywhere. So how can we use this phage display, how it works? First of all, we have the capsid of a phage or any virus actually, and these capsids, they contain usually two types of proteins: major proteins that form the majority of the proteins, and minor proteins that are presented in the smaller amounts. So, if we put a gene that codes a protein of interest near the minor protein, our protein of interest, these molecules, will be expressed, displayed on the phage particle near the minor proteins. If we put the gene near our gene of interest, near the gene that codes the major protein, these proteins of interest will be expressed on the surface where are the major proteins. So the difference is just in the amount of the molecule of interest that we can receive with this phage display system. So phage display means that the phage displays some new molecules on its surface, and we can take them off and use them in biotechnology and medicine as we wish. So if we have some pool of genes, some genome, and we just put them with the help of genetic engineering into the phage particles, we receive a lot of phage particles that have different genes and express different proteins on their capsids, on their surfaces. And this is called a sort of library. So there is a pool of phages that have a pool of genes and express different proteins. But we cannot work with this library because it doesn't have any order. They're like a lot of different proteins. So we have to pull out our protein of interest or just make a sort of order, organize these proteins in sort of groups. How can we do it? We can use some biological substances that will absorb our protein of interest, for example, and then we will wash away all the other phage particles with different proteins, and only these proteins of interest will remain. Then we just take them off again, so they are free, and the only thing we have to do is to put these phages inside the cell, and this cell will work as a factory to produce more phage particles that contain our proteins of interest. Then we'll simply take these proteins off the phages and we will use them everywhere we want to use them. For example, this phage display is widely used in the construction of biosensors because we cannot construct a biosensor with only one molecule. We need a lot of copies of one and the same molecule. And the easiest, the cheapest, and the very stable way to receive a lot of copies of any protein or peptide molecule is to use phage display. So, as we go on, we turn to biosensors, and we have to just say a few words. What is a biosensor? A biosensor is a device that uses a biological and a mechanical component. Biological component is a sensitive surface with some substances, biological substances. These can be any molecules like nucleic acids, proteins, phage particles, viruses, bacterial cells, part of the cells or the whole cells, and even eukaryotic tissues can be used on the sensitive surface. When the sensitive surface interacts with some chemical or biological or even physical factors from the outside, it creates this interaction creates a signal, and this biological signal is turned with the transducer into the sort of answer, quantitative or qualitative result that we can analyze. So, as I already said, phages can also be used in the construction of biosensors. Here you can see the sensitive surface with the phages on it and the cell that bounds to these phages. It means that we can receive either qualitative result, that means that yes, the cell is present in the sample, or no, there are no bacterial cells in the sample. Or the quantitative result, kind of like count how many cells are present in the sample. For example, 100 cells are present in the 1 milliliter of sample. This is the quantitative result. And so, biosensors, including biosensors based on the phages, can be used in medicine to analyze human samples, in food production to analyze the quality of food, also in ecology to analyze the quality, the state of soil, water, also the contamination with pathogenic bacteria or non-pathogenic bacteria, but anyway the contamination with bacteria. Now can be studied with these sensors based on the bacteriophages, and we will have these results using these phages. So, as I already said, both types of phages can be used in the construction of biosensors. These can be classical phages with head, tail, and fibers, and filamentous phages that express a larger amount of new proteins. The phage display system is better to be performed on these phages in particular. And what new approaches are used, including phages, to construct biosensors and to use these phages as sensors? Well, there are a lot of approaches, and some of them are really very interesting, like Imagine probe, Imagine with phages. Imagine method means that we can study not the sample taken out of the organism, but study something exactly in the organism using phages. Because these phages can be loaded with fluorescent dye, for example, and if the patient takes these phages in, they target some cells or tissues and show us, for example, where exactly is the inflammation, what bacteria causes the inflammation. And this is performed within the organism. We don't have to take samples, bring them to laboratory and study them. It can be performed with the patient, right, like real-time diagnostics. Also, phages can be used in biosensors for bacteria immobilization. The principle we already talked about. So we have the phages on the surface and they just bound the bacterial cells. Also, phages can be used to enhance the signal, because one molecule gives a smaller signal, of course, and the more molecules we can detect, the bigger will be the signal, and the better will be the result that we receive with the use of such biosensors. Well, also these sensors can be used to detect bacteria, like already given the information not about the bacteria present in the sample, but already given the information about what bacteria are present in the sample, because phages interact with bacteria very specifically. And we can even classify or type these bacteria, for example, that cause the disease. So, as I already said, phages can be used to form sensitive layers of biosensors, like icosahedral phages and filamentous phages, and also they can form electrode materials. These sensitive layers and electrodes, they just differ in the form of the analytical part of the biosensor. And as I already said, just to repeat it one more time, different biological molecules can be used in the construction of biosensors. These can be nucleic acids, antibodies, And also phages can be used. We can use wild-type phages, the phages that are present in nature, to, for example, detect bacteria. But it's better to use genetically engineered phages so that they won't cause the lysis of the cell. Because if we have wild-type phages, they bound to bacteria, we have the signal, here's the bacterial cell. But if they cause the lysis of this cell, the cell disappears and we lose the signal. So these results will be unstable, but using genetically engineered phages that, for example, simply bound the bacteria, will make the signal more stable. Also, only the we can also use only, for example, tail fibers or receptors of phages just to bound bacteria, but not to destroy them. But also genetically engineered phages can be used, for example, reporter phages. Reporter phages are very interesting because they are genetically changed so that they carry some gene that starts to express within the cell and produce some sort of product is produced when the cell is infected with this phage. And with the production of this product, we can tell again, we can tell what bacteria is detected with this phage. We can tell how the infectious process goes on. So we can just make a real-time research while these interactions of reporter phages with bacteria cells. So, as I already said, the only necessary thing to use phages in biosensors is to immobilize them on the sensitive surface. If we just simply use random immobilization with physical or chemical factors, phages can be immobilized with immobilized in a chaotic order. It means that some of them are turned upwards with their tail fibers and some of them are just turned downwards to the sensitive surface. It means that, for example, this phage particle won't interact with the cell. And this is wrong. We cannot use this approach if we want to construct a good biosensor. So we need, for example, a charge-directed immobilization so that all the phage particles will be in one order, giving us their receptors turned upwards so that they will interact with bacterial cells. And here is the simple scheme of how this biosensor works, for example, in the laboratory. So we have the sensitive surface. We put some, for example, chemical or biological substances so that we can bound our phage particles in the right order. Then we use this sensitive surface of biosensor with phages. We use them to analyze the sample, and if the sample contains some sort of bacteria, they specifically bound to these phages, and we already have the result. Whether there is interaction, what bacterial cells interact, how many bacterial cells are present in the sample. And this experiment will take from few minutes to few hours to be performed, because the classical approach to analyze whether the bacteria are present in some kind of medical or biological sample, it will take few days with classical approach to create nutritious media, to put bacteria on it, to grow them, to study the interaction, to type these bacteria. So it will take within several days. But this approach using biosensors will take just from few minutes to few hours to give us the result. So it's much quicker, it's much better, and it's very important when we talk about diagnostics, especially medical diagnostics. So time really matters. And so, just a glimpse at the biocontrol. These biosensors and phages themselves can be used in biocontrol. Biocontrol means that we use some biological systems, biological approaches, biological substances or organisms to fight some biological problem, fight another biological organism, like fighting bacteria using phages. This will be phage biocontrol. Phages are widely used because they specifically bound to bacteria, because they are very stable in the environment. For example, if we use some chemical substances, they can be inactivated in the environment. But phages won't be inactivated because they are part of the environment. They are absolutely biological organisms. So, and phages can be used to fight bacteria in food products, in soil, water, and to, well, in medical sphere to, for example, cure diseases, and we were already talking about this. And of course, biocontrol with phages, this interaction between phage and bacteria will be influenced by physical structure of the component that we work with: food, soil, water, human samples, and also will be influenced with environmental conditions like temperature and the reaction of the environment. But all these factors can be encountered and improved with biotechnological approaches. So, we were already talking about phage therapy, and this is part of biocontrol because we can use phage therapy, for example, for domestic animals, and this will be absolutely ecological approach. We can also use phages for disinfection of equipment and different surfaces, and this will be again biological approach, ecological approach, because there is no need to use chemical compounds, for example, to disinfect something. Also, phages are widely used to prolong the freshness or shelf life of products. If we talk about fresh products or ready-to-use products, so this is actually possible to use phages. The examples: we can use phages, for example, to keep fresh milk fresh for longer time. It means that if we just use phages to reduce all the bacteria from milk, it can make possible to keep this milk fresh for a few days. It won't be spoiled because there will be no bacteria that cause spoiling of this milk. And if we talk about cheese, cheese can be saved for longer time because this is a ready-to-use product. But also, if there will be present some sort of bacteria that cause food spoiling, the cheese also will be spoiled. And we can use phages to eliminate these bacteria off the cheese, for example. It can be sausage, it can be some sort of meat, any type of food. And without these bacteria that cause spoiling, the shelf life of this product will also be prolonged. Without any chemical compounds used to conserve this product, so it will be healthier, it will be better for us because this food will be healthier, more natural, and better to use. So this is just a glimpse of how phages can be used in biocontrol. I believe that we will talk about it on our next and final meeting in more detail. In more details. So, and as for today, I think that this will be enough. Thank you for your attention, and see you next time. Goodbye.